Hello all. In this video, we are going to discuss about bacterial staining. Bacterial staining is one of the basic techniques which is used in microbiology labs. It is used in the visualization of bacteria in microscopy. Here, we will discuss about why and how we are doing the bacterial staining technique. First, we have to address the question, why do we do the staining? It is because microorganisms like bacteria are microscopic and cannot be observed with our naked eyes. And also, we always suspend the microbes in certain aqueous media, and therefore, it is colorless and transparent in the aqueous medium if we do not stain them with a particular stain or dye. Thus, there will not be any contrast between the microorganism and the aqueous media we are using. They will be looking alike, hence their visualization will be very difficult. Also, the refractive index of the microorganism and the aqueous media are very low and almost the same. Due to these reasons, we use the staining technique for a better visualization. Now we will talk about the objectives of doing bacterial staining. First is something we have already discussed, that is for the visualization of microorganism in a very good way, because when it is not stained, microorganisms are very difficult to visualize. The second objective is that through staining we can attain a good visualization through which we can study their morphological properties and group them accordingly for many purposes, like that for diagnosis of certain conditions. And also, in the case of bacteria, the microscopy that we employ is a light microscopy. Now, what is a stain or a dye? We already said that bacterial staining is done with a stain or a dye. Now we can look into what the stain or dye is chemically. The stain or dye is usually an organic compound and it has three components. First component is a benzene ring, which is the organic colorless solvent part of the stain. Then the second component is a chromophore group, which imparts color to the benzene ring. The third part is an oxochrome group, which conveys ionization property to the chromogen part, enabling salt formation, and also it binds to the tissues or fibers that we are observing. The benzene and chromophore parts together is called the chromogen portion of the stain, and when oxochrome part is added to the chromogen part, together it is called the complete stain. Neither the chromogen part nor the oxochrome part cannot be considered as a complete stain. Both these parts are required to call it a complete stain. Now, what is the difference between using the terms stain and dye? Mostly these terms are used interchangeably, but there is slight difference in using these two terms. Usually, stain is used to visualize biological specimens and dye is used to visualize non-biological samples and for general purposes, like dyes are used in textile industry and all those, while stain is used only to visualize biological specimens. And also, stain is in purified form whereas dye is a very crude form. And also, for the visualization of bacteria commonly, the stain term is used more than dye due to these reasons. Now, there is another term we have to be familiar with, that is a mordant. What is mordant? Mordant is a substance that is used in staining that enhances the affinity of the stain to the cell or tissue that we are observing, but the mordant itself has no affinity or it has a very low affinity. Usually, the chromogen part of the stain is electrically charged, and also this electric charge found on this chromogen and that in the cellular components that we are observing enables the stain to bind to cellular components. Based on this property, stains can be of three types, that is acidic, basic or neutral. Here we are going to discuss each one of these in detail. First, acidic dyes. Chemically, acidic dyes are sodium, potassium, calcium or ammonium salts of colored acids upon ionization, which means that when chromogen group is ionized, if it is giving a negatively charged chromogen, they are called acidic dyes. Whereas basic dyes are chemically chloride or sulfate salts of colored bases upon ionization, which means that when chromogen is ionized, if it is giving a positive charge chromogen, they are basic dyes. Now we can discuss a few points about acidic and basic dyes. First, coming to acidic dyes, they are anionic molecules and their chromogen part has a negative charge when they are ionized. And since they are negatively charged chromogens, they have strong affinity for positively charged cellular constituents like proteins. And the cellular constituents can readily bind to this negatively charged chromogen and take up its color as they are oppositely charged. And examples for acidic dyes include picric acid and nigrosin. Now, basic dyes. Basic dyes are cationic molecules and upon ionization, their chromogen carries a positive charge. Since the chromogen is positive charge, they are strongly attracted to negatively charged cellular constituents like nucleic acids, and also these negatively charged cellular constituents can readily bind to the positively charged chromogen and take up its color. And examples for basic dyes include methylene blue. Now, neutral dyes. Neutral dyes imparts both positive and negative charge on the chromophore after dissociation or ionization. Example for neutral dye include Leishman stain. Bacterial staining in particular commonly employs basic dyes. They rarely or mostly never use acidic dyes. This is because the bacterial surface has a negative charge on them, and this negative charge will repel acidic dyes which are also negatively charged, and also this will make it impossible for the stain to penetrate into the cell. This is why negative dyes or basic dyes are used in bacterial staining. And the reason for negative charge on bacterial surface is different for gram-positive bacteria and gram-negative bacteria. The presence of teichoic acid, that is having a negatively charged phosphate group linked to either peptidoglycan or underlying plasma membrane, is the reason for negative charge on bacterial surface of gram-positive bacteria, whereas in gram-negative bacteria the presence of lipopolysaccharides on the outer covering of the bacterial surface is the reason for negative charge. Now we can discuss about types of staining techniques. Staining techniques can be classified mainly into two: simple staining and differential staining. Simple staining is when we use a single stain only for the visualization of microorganisms, whereas differential staining is when we use two contrasting stains for the visualization. The simple and differential staining techniques can be connected to their purpose as well. Simple staining is used for the visualization of morphological shapes like cocci and bacilli and arrangements like chain, clusters, tetrads, etc. Whereas differential staining is used for the visualization of more specific bacterial structures like flagella stain, spore stain, nuclear stain, etc. and for the separation of bacteria into groups. For example, like gram stain or acid fast stain. Here it is very visible that differential stain uses two contrasting stains for making a contrast between the specific bacterial structure we are observing from the rest of the cell. We have to discuss each of the staining method in detail, and those will be discussed in the coming videos. Thank you for watching.